Okay, biology students, we have reached our final lesson in our genetics unit. And in today's lesson, we're going to focus on how biotechnology utilizes living organisms to develop products and technologies for various applications like agriculture, healthcare, and even forensics. All right, so if you're in class with me, we're going to watch this video that can be searched on YouTube about CRISPR. And CRISPR is a form of biotechnology. And remember, biotechnology is using technology bridged with biology, essentially to solve a human problem. And a great example of this is the production of vaccines. So vaccines, of course, are applicable to all of our lives, and biotechnology is a way of producing these vaccines. Now, in biology, we focus on the use of biotechnology to manipulate an organism's genome. So an organism's genome, when we say this, we're referring to an organism's genetic information. And so when we use biotechnology to manipulate an organism's genome, we call this genetic engineering. Now, today we're going to research several different aspects of genetic engineering, and we're going to discover their implications on humans and the world around us. And there are tons of different examples and ways that genetic engineering is being used today, but we're going to focus on three categories: agriculture, medicine, and forensics. Now, we have already discussed the fact that DNA can be manipulated, and scientists use different techniques to do this. Now, scientists can't manipulate DNA with their hands, but they can use chemicals and computers and even bacteria to manipulate an organism's DNA. So the manipulation of DNA, again, is called genetic engineering, and it has resulted in advancements in three categories, three specific categories that we're going to discuss in this particular lesson. So again, medicine, forensics, and agriculture, and we're just going to kind of go through each of these. So just an overview of what we're going to discuss today. For medical purposes, genetic engineering is used to produce medicines and vaccines like insulin. We can use genetic engineering to modify lab organisms to make them easier to study. In agriculture, genetic engineering is being used all over the world to improve crops. We can make crops that are resistant to diseases, pest, frost. We can make crops that provide better nourishment. We can make crops that have certain traits like a certain sweetness. We can make watermelons that don't have seeds. The possibilities are really endless with using genetic engineering in the field of agriculture. We can also use genetic engineering to help solve crimes. So in forensics, we can take unknown DNA samples found at a crime scene, and we can process them through a process called gel electrophoresis to get sort of a barcode or a DNA fingerprint. And then we can match that DNA fingerprint to different suspects. And if we get a match, we know the source of the DNA, which is really cool. All right, so let's start with biotechnology in forensics. So one type of genetic engineering is called DNA fingerprinting, not to be confused with actual fingerprinting. So DNA fingerprinting has nothing to do with fingerprints. DNA fingerprinting is like a barcode, if you will, that you get through a process called gel electrophoresis. So we can take crime scene DNA, run it through a process known as gel electrophoresis, and we can get like a genetic profile of a person or the donor of the DNA. And then we can use that to compare to suspect DNA samples. And if there's a match, that can help forensic investigators essentially solve a crime. Earlier when I mentioned gel electrophoresis, it is a complicated process, but in a nutshell, it's just the process of creating a DNA profile. And so in the bottom right-hand corner, you can see this is an example of a DNA profile. So we have collected crime scene DNA, and then we have collected the DNA from 3 suspects that we believe might have something to do with this crime. And you can see here that we've run a genetic profile for each of the suspects and the crime scene DNA. Again, this is done through the process of gel electrophoresis. And then we can match these fragments, and we can see here that suspect 2 matches perfectly with the chromosomal DNA. Now, there are several other applications of DNA fingerprinting. Of course, it's used in forensics, but it can also be used to determine paternity, and it also can help identify victims. And so if you're familiar with 9/11, when terrorists attacked the World Trade Center, they had to use DNA fingerprinting to help identify some of the victims. And they're actually still using DNA fingerprinting today to help identify some of those victims. All right, so blood, hair, cheek cells, all of these can provide samples of DNA that can be used to create a DNA profile or a DNA fingerprint. Again, here's an example of how DNA fingerprinting can be used in forensics. So if you look on the left side of the screen, we have a blood stain at a crime scene, and then we have 4 possible suspects, so Bob and Sue and John and Lisa. And if we match these fragments, notice some of the DNA fragments do match some of the individuals, even though they're not an exact match. But if we compare the bloodstain DNA profile to John, we can see that these fragments match up perfectly. And so in this simplified example, John would be the donor of the blood stain left at the crime scene. Also, we mentioned earlier that paternity can be used, or DNA fingerprinting can be used to determine parentage. So remember, a child's alleles, 50% of its DNA comes from mom, 50% comes from the biological father. So using this idea, we can get a child's DNA, and we can get the mother's DNA, and then we can get suspected potential fathers of the child, and we can sort of make a comparison to figure out which male, in this case, is most likely the father of the child. So I'm just going to go down and look at these fragments here. So this fragment here matches mom, which is not going to be very helpful to us. However, this fragment and this fragment, neither of these match mom. So that means this has to match the biological father. And so we can see here how male 1 would be most likely the father of this child, because these 2 fragments do match up. I'm skipping through some of this just for time's sake. All right, so we just talked about biotechnology's use in forensics, but biotechnology can also be used in medicine. We can create vaccines. We can cure some diseases. We can create treatments for disorders and diseases, and even some cancers using genetic engineering, which is super cool. So the main goal of the Human Genome Project, which was, it got scientists to come together to basically map out our genes. This happened in 2003, and so we were able to look at for the first time, look at a person's complete genetic profile. And so the main goal of this project was to find out where certain genes are, so that we can identify genetic disorders, and then, of course, ultimately treat those genetic disorders. So from that has come a ton of medicines and vaccines and treatments for a number of different genetic diseases and disorders. Now, today, researchers use something called recombinant DNA technology to analyze genetic changes. So basically, researchers are able to cut and splice, kind of cut and paste, and insert modified DNA molecules from different species into bacteria and some other types of cells. And the reason why they use bacteria is because the bacteria rapidly replicate and divide. And so these cells copy the foreign DNA right along with their own DNA. Again, a great example of using bacteria to help solve a problem is through the production of insulin. We have discussed before bacterial plasmids, but remember, bacteria are prokaryotic cells, and their DNA does not live in a nucleus, but we have bacterial plasmids that contain DNA. And so scientists are able to remove the bacterial plasmids and cut it. They can then insert a desired human gene into this bacterial plasmid and then take the modified plasmid, which at this point is called recombinant DNA, and reinsert it into the bacterial cell. And the bacterial cell will produce whatever protein the inserted gene codes for, and it can do this in mass quantities. And again, this is how insulin is made. Now, when DNA from one organism is inserted into the original DNA of another organism, we have a name for the new DNA, and it's called recombinant DNA. And so you can see here in the picture, we have a bacterial plasmid. We have cut out the old gene. We have replaced it with a desired gene. And so at that point, we then have recombinant DNA. And then when that DNA is inserted into an organism, we call it a transgenic organism. All right, biotechnology is also being used in agriculture. So biotechnology provides farmers with tools that can help make production cheaper and more manageable. For example, some biotechnology crops can be engineered to tolerate different herbicides. They can make, of course, this can make weed control simpler, more efficient. Other crops have been engineered to be resistant to specific plant diseases and insect pests. This can make pest control more reliable and effective, and can decrease the use of synthetic pesticides, which helps the environment. Plant biologists have used DNA technology to produce plants with many desirable traits, and we call these organisms GMOs, genetically modified organisms. There's a lot of debate that surrounds GMOs, but my guess is if you go to your local grocery store, most of the produce that is in the produce section is a product of GMOs. One of my favorite GMO products at the current time is the cotton candy flavored grapes. If you've not tried those, they are delicious. They are GMO, so somebody has essentially figured out a way to create grapes that taste like cotton candy. This chart shows some examples of how biotechnology is used in agriculture. You can just take a look at this list. One of the coolest uses is in countries where there is not a lot of nutrients. We can actually, in the United States, we have been able to produce crops that have the nutrients that some of these third world countries are lacking. So we can have rice that has added vitamin A, or hepatitis B protection added in bananas. And then we can send those over to other countries to be grown, which I think is super cool. Right now, we know that genetic engineering provides us with a ton of benefits, right? So we've talked about how we can solve crimes using biotechnology and genetic engineering. We can cure diseases. We can treat diseases. We can grow crops with favorable traits, right? It seems wonderful, and it is. But with every benefit, there are associated risks. So it's important that we kind of balance these out, and we also talk about the risk of genetic engineering. Bottom line, genetic engineering is a newer technology, and so we don't really know the long-term implications because it hasn't been studied long-term. We do know that in agriculture, when we use genetic engineering and GMOs, this does decrease genetic diversity. And we're going to talk about that more when we get to our evolution unit. There's also a risk of unintentional mutations and biological resistance that comes with that. Again, we're going to talk about this more when we get to evolution. There's also some safety questions that come up. We don't really know if GMOs are safe. We don't really know if certain vaccines are safe because, again, these are newer technologies and the research is still being done in some of these areas. There's also some ethical questions, like where do you draw the line? If we can alter somebody's DNA to create traits that are admirable or desired. If we can do this in crops and produce, can we also do this in humans? And where do we draw the line with that? So it does bring up a lot of questions, and it's important to just kind of weigh the benefits with the risk. This ends our last lesson of our genetics unit, and I will see you in our next unit, which is going to be heredity.